Now I'd like to call upon a professor, a doctor, currently working as Associate Professor in St. Mary's College of Pharmacy, Secunderabad, Telangana, India. He completed B.Pharm from Dr. MGR Medical University, M.Pharm from Jadavpur University, Kolkata, and PhD from University of North Bengal in the year 2013. He received the prestigious DBT Overseas Visiting Scientist Award for the year 2015 and earned postdoctorate from Cancer Research Laboratory, Augusta University, USA. He has 17 years teaching experience, including 8 years research experience. He guided 25 PG students and published 35 research papers in national and international journal of repute when he serviced as Associate Professor in Himalayan Pharmacy Institute, Sikkim. He visited UK, US and France to explore his research work on breast cancer chemotherapeutics. He delivered the invited lectures in Paris, France, Anna University, Tamil Nadu, Kalasilingam University, Tamil Nadu. He completed 2 AICT projects under RPS scheme. His major work related with drug synthesis and biological screening, particularly anti-cancer studies. Now he is gearing his research towards stem cell targeted breast cancer chemotherapeutics. He is the life member of Indian Association for Cancer Research and Association of Chemistry Teachers of India and also Indian Science Congress Association. Sikkim University conferred Best Teacher Award in the year 2011 for his outstanding contribution towards teaching in northeastern states, particularly in Sikkim. He is none other than Dr. G. Mariappan.
Respected President, Dr. Nageshwar Rao, the Multipurpose Awareness Society, Madam Imam from Egypt, Madam Minaja from Malaysia, and Mr. Mohitullah from this MAS, and my students and all the delegates, good afternoon to all. So continuously we are observing the various lectures. So in addition to that, so we know very well about these pandemic situations, and in this pandemic how much our people are affected. So in these conditions it is wise to discuss about these corona vaccines, and it is wise to create the awareness amongst the people, because still now several people are afraid to take the vaccines. So from the medical perspective, from the research perspective, how these vaccines are discovered and how much really it is useful for the people of our society. So if you see the total world population, almost 760 crore. In these 760 crore, 50% of the populations are in India as well as in China. And China is the leader class. Keeping 140 crore population in our country, vaccinating all the people is not a matter of joke. But still our country have achieved all these things. So we vaccinated almost 80% of the people, except the children and very, very elders, only we could not do that. Other than that, 80% of the population of our countrymen is the vaccinated. Now, according to the WHO report, 2.67 billion people are affected by this coronavirus. Sir was telling India is the country and politicians are not looking the poorest poor. Yes, it is true. We have to consider all these things, because we cannot ignore any life. It may be human being, or it may be ant, or it may be the housefly. Life is life in this planet. There is no excuse. Every creature has a life to live. And this pandemic situation, how much the virus killed the people, more than the virus, this poverty killed the people. One incident which I have seen, which is not wise to see again and again also in the YouTube or in the Facebook. It happened in Tamil Nadu. One person was walking in the road. Just he is walking. He's walking. So he is coming, he is looking front and back. Nothing is there. So one truck came. Truck means it is more than, I think, 15 tire. Truck came and truck was waiting for the signal to go. Within fraction of the second, he went inside the tire and finished. Life is over. Within fraction of the second. It came in the YouTube also. The reason for his death or suicide is that no money to eat the food. Because during this corona time, everything is closed. College is closed, school is closed, job, nothing. Everybody is inside the home. Understood? So how much this, but still the humanity exists in our society, the scientists, the people. So government is also having lot of pressure to bring this vaccine to the market. And coronavirus, the first wave, again this second wave, how fast it killed the people, so we know that. So in these current situations, it is wise to discuss about these corona vaccines, and it is wise to create the awareness amongst the people. So I am going to give the talk about these corona vaccines and how these vaccines are discovered and how much it is useful for us. Almost 5.3 crore people till yesterday died because of this corona worldwide. 2.67 billion people are affected by this coronavirus. So it is a mandate, so we need to know about these vaccines. And Telangana is the state in India, really what Dr. APJ Abdul Kalam dreamed. So we need to build our nation. So Telangana is the state. The Abdul Kalam said once in the Indian Science Congress meeting that we need to be very much self-reliant. 2020, Abdul Kalam was not there, but the scientists, the technocrats, the people of the Telangana really made Abdul Kalam true. I really salute the people of the Telangana and the scientists of the Telangana and the microbiologists of the Telangana. They have developed this indigenous vaccine in Bharat Biotech, Hyderabad, and supplied all over India. Even though if no country given the vaccine to us, from US or from UK, there is no problem for us. The Telangana, the Hyderabad, the Bharat Biotech have the capacity to produce the vaccine and ready to vaccinate all over the people of the India. So really a great salute to the scientists of the Telangana. Now, let us enter into this vaccine platform. So now, how these vaccines are really discovered? So we are talking about vaccines, these corona vaccines, this Covaxin or Covishield. So we know very well about this coronavirus. It is RNA virus. Because of this virus, because of the droplets from our nasal secretions or face-to-face communications and all, through air the virus spreads. And it affects the people, it will affect our respiratory system. As a result, pneumonia, the final stage is death. So everybody knows about this mechanism of the disease, how the disease spreads. Fine. So now the synthetic molecules or the natural products are having now very limited role to save the life of the patient, because it is a virus which replicates very quickly and we need to save the life of the patient. So the synthetic, several antiviral drugs were attempted, but the result was not that much satisfactory. So several natural products also attempted, the result was not so much satisfactory. So the only option was the vaccine, because we need to boost up our immune system which can control this virus. So that with that objective, our government of India and the whole world, so everybody, in a synchronized manner, they involved in this vaccine research. As a result, within 1 year we have got the vaccine. Otherwise, a normal vaccine, it will take at least 4 years. But since it is a global problem, so everything in a beautiful manner, so they got these vaccines. So now, how many types of the vaccines are there? We can develop the vaccine from the inactivated virus. We can develop the vaccine by using the attenuated virus or the viral vector. By using that also we can develop the vaccines. DNA vaccines are there, RNA vaccines are there, subunit viral protein vaccines are there, or virus-like particle vaccines are there. Like that, the vaccines are several types. According to the necessity, according to the need, the pharma companies, they will adopt different methodology to develop the vaccines. Now, the best example for these live attenuated vaccines, these measles, mumps, rubella or yellow fever, the vaccines for all these things, we can use the live attenuated virus. That means what? We will not kill the virus, but the virulence of the virus or the pathogenicity of the virus can be controlled. So if we control this virus, then the virus have no replicating capacity. Our main point is that once if the virus goes inside the body, automatically it will replicate. If we keep the virus on this table, nothing will happen. It will remain just like a sodium chloride crystals. The same virus, if you inject into our body, it will start to replicate. So that we are telling that it is virus particle. Just like a solid particle, it's a particle. So the capacity of the virus is such that when the problem happens, when it replicates. If it doesn't replicate, there is no point of the problem. So, but our immune cells, our biological system such that just we have to cheat the virus in such a way that our body will recognize that foreign particles, automatically it will produce antibodies. Virus can be controlled mainly by vaccines. The synthetic molecules or the natural molecules or the marine products have very, very limited role because the replication is such. So in one way we can prepare these live attenuated vaccines. In another way we can prepare the inactivated virus vaccines. Means we will kill the virus. Virus will be completely dead virus. One is live virus, another one is dead virus. The best example, Covaxin. The virus is completely inactivated. The virus is completely inactivated. There is a mechanism to inactivate the virus, but virus will remain. The virus particle, everything will remain, but virus will die. Die means what? It cannot replicate. That is the point, because we need to boost up our immune system. Vector-based vaccine, the best example is Covishield vaccine, which is developed in Serum Institute with the support of the Oxford University. So the Covaxin is absolutely designed only by our Indian scientists. But the Covishield, Indian scientist with the scientist from the Oxford University. So that always we are comparing this Covishield with Covaxin. But both are same. The DNA vaccines. So the same, we have to take particular segment of the DNA, we have to inject into the virus, and we have to produce the vaccines. So this is DNA vaccines. mRNA vaccines, whatever the vaccine is coming from US, almost all are mRNA vaccines, like this Pfizer or Moderna. These are the mRNA vaccines, because different companies have the ideology. Protein subunit vaccines, the best example for influenza vaccines, because always we cannot handle the complete virus. We need to take one particular segment of the virus and we will inject this thing into another vector and we can produce the subunit. That subunit will activate your immune system. Virus like particle vaccines, the same thing. The best example is hepatitis B vaccines. Now we can see always in India these two vaccines are very popular because we are not taking the Russian vaccine or we are not taking the US. When we are having our own products, so we don't need to immediately go to US or UK. First let us be individual. Let us be self-reliant. What our forefather Abdul Kalam said. We need to be first self-reliant. So when we are able to develop the vaccines by our own, when we are able to give the vaccines to our people, let us achieve that. So this Covishield, Oxford University and AstraZeneca and Serum Institute of India Private Limited, Pune. So the Bharat Biotech, which is well known, you know that it is in Hyderabad. So these are the two vaccines which is available and people also used. Now the Sputnik vaccines, from the name itself you can understand it is from Russia. So they have used different technology to develop this vaccine. These are the vector-based vaccines. They have used two types of the vector to design these vaccines, which is also, the efficacy is also very good. And these Moderna vaccines, which is from United States, so they have used, this is mRNA vaccines. So one is live virus, another one is dead virus, and one is vector based, another one is mRNA based. So these are different types of vaccines, but ultimately their aim is to instruct or to boost up the immune system to generate the antibodies which can encounter the virus. So now, the same thing, the Pfizer, it is also mRNA-based COVID-19 vaccine jointly developed by an American pharmaceutical company, Pfizer, and German biotechnology company, so that the name is BioNTech. Now, how these covaccines now is developed, what are the chemical compositions, because these are playing, vaccines directly we cannot inject. So when we come to the pharmaceutical point of view, So we need to add several ingredients so that the vaccines can be stable for a certain period of time, because we cannot develop the vaccines and today immediately we cannot inject. We need to develop the vaccine, we need to store the vaccines, we need to transport from one place to another place, particularly to the African countries. African countries are completely relying upon India because we are the big exporters of medicines to the African countries or the Middle East. So in any vaccines, number one virus particle or DNA or RNA or this live virus or dead virus must be there, that we can say antigen, and aluminum adjuvant should be there because to increase the adsorption property, because these vaccines should bind with the surface of the virus. So we know that the virus has these spike-like proteins. So for that, this aluminum adjuvant will be very much useful. The vaccine should go very close to the virus so that there can be a binding. Then there are certain excipients like buffer solutions or sodium chloride surfactant stabilizers, because these are the liquid formulations. Vaccines are liquid formulations, it needs more stability and it should be stored in very low temperature. So during these storage conditions and all, the stability of the vaccine should not be spoiled, so that they will add all these things and they will maintain, and the vaccines always will be stored in 0 degrees Celsius or even less than that. Then there is certain the formulation point of view, which pharmaceutical company generally don't disclose, because for their patent and all they will. At last it will come to the market. Now for your the information, we can say that this the whole virion inactivated virus, first composition aluminum hydroxide gel for the as adsorption on antigen, then imidazoquinolinone as an adjuvant. They will induce this TLR signaling in the modulation of the innate immunity as well as adaptive immunity. So what is the use of this thing we can see, and 2-phenoxyethanol we will use as an antimicrobial and preservative, and phosphate buffer saline we will use for the maintaining the pH and all these things. So these are the if you take any vaccines, only virus will not be there, including virus, all these composition will be there. Then only it will come to the market. They will manufacture in bulk productions and it will be given to the patient. How these covaccines works? Okay, virus is there in that covaccines. How they are inactivating the live virus into because live virus if you inject finished, the next minute we will not be available. So the live virus will be converted into inactive virus by using this the beta propiolactone. It is a chemical substance which will inactivate the genome. You see this virus, this is a live virus, spike protein. Within that the RNA will be there. This RNA has the replicating capacity, but here you can see the color code, it is almost dead. So this RNA cannot replicate. Once if you inject this virus, simply it will remain in the virus. But at the same time, since it is a foreign body to our body, so because of these spike proteins, your immune cells will be boosted and activated. Once if the immune cells are activated, immediately it will generate immunoglobulin G, otherwise called as antibody. That antibody will counteract the virus if any virus goes inside. So in this way, the best example is these polio vaccines, these rabies, hepatitis, all are the inactivated virus vaccines only. So after the inactivating the virus, this virus will bind with the antigen presenting cell. So there is a receptor, it will go engulfing the virus. Once the virus is engulfed, what will happen? This protein particle only will be coming out of the virus. So this protein particle again will be expressed presenting the virus fragment protein. So these protein particles will activate the T lymphocyte, or we can say T cells, and these T cells will be activated. The activated T helper cells will activate B lymphocytes. All of our antibodies will be released from the B lymphocytes. So when the B lymphocyte is activated, it will release the antibodies, immunoglobulin, particularly immunoglobulin G, and that immunoglobulin G will counteract the virus, attacking the live coronavirus. So wherever these spike proteins are there, all the spike proteins will be covered by these antibodies. As a result, this virus cannot replicate. So in this way, this covaccine acts. Almost all the vaccines mechanism is in the same way. By any means, whatever the way they are manufactured, they have to activate the the B lymphocytes, and B lymphocytes will release the antibodies. Classical vaccines, if you prepare a vaccine in a normal conditions, it will take almost 2 to 3 years or even more than that. But during this pandemic time, all the pharmaceutical companies very quickly they have activated the process in the preclinical stage, phase 1, phase 2, phase 3. As a result, the emergency use authorization, the government of India, the drug controller of India, they have given the emergency use authorization, so quickly it came to the market. So now the Covishield. How the Covishield vaccine is not the dead virus vaccine, it is the replication deficient chimpanzee adenovirus vector vaccine. This is vector based vaccine. So that means we can see that the following administration, the genetic material part of the coronavirus is expressed, which stimulates an immune response. You see the composition of this the Covishield vaccines. So these are the composition. L-histidine, Covaxin, some composition is there. Covishield, other composition is there, because their starting material is different. Magnesium chloride hydrate as a stabilizer, polysorbate 8, emulsifier, ethanol preservative, sucrose cryostabilizer. Like that, there are several compositions to make the vaccine stable. How they are making this vector? They are taking the chimpanzee adenovirus. This virus they are modifying. Now you see the color of this virus. It is just color code just to differentiate the original virus from the modified virus. Unable to cause disease. That means it cannot replicate. So it doesn't have the capacity to replicate. This is entering into the, and the spike protein. They are taking the coronavirus, and from that coronavirus the particular the gene which can code this spike protein is isolated, and they are sequencing that particular gene, and that gene is entering into this the modified virus. So as a result, so this modified virus doesn't have capacity to replicate, but it has the capacity to boost up the immune system. As a result, the antibodies will be produced. So in this way, this the Oxford vaccine is working. Now the same thing, if you see the Russian vaccine, Sputnik 5 vaccine discovery, they are using two viral vector. One is this vector AD26, another one is 5. So the same thing, they are taking one particular segment, the gene which codes for this spike protein. Spike protein is very, very important for this coronavirus. Because of this thing only, the binding takes place. So as a result, so they are injecting this genetic material into vector 1 as well as vector 2. So 2 doses are giving. First dose, I will. So anyway, so you can see this mRNA vaccines, the nucleic acids vaccines, same thing, DNA vaccines as well as RNA vaccines. mRNA vaccines platform, you can see that. And always we don't compare our Indian vaccines. We will fight amongst ourselves only. The Covaxin is very great or Covishield is very great. We don't compare Russian vaccine with US vaccines. So we need to always have this the feelings that we are great and we are Indians, we have the capacity. So if you see the efficacy point of view also, this Bharat Biotech is 80% and our Moderna is 94, Oxford like that. So our Covaxin is also coming within this line. It is also not lesser than that. So let us also feel very great because our country has developed this vaccine in this pandemic time. If we don't have our own vaccines, it is very difficult for our country to save all of our people because our people is more in this world. Let us feel proud. So thank you very much for your